In laboratories around the world, what was once science fiction is becoming reality. We are deciphering a hidden code locked within each of us. We now have the tools to explore the great mysteries of life. With amazing precision, a scientist captures the first moments of life and alters the destiny of a simple organism. This strange creature may give him clues to how life begins. We still wonder just who is related to whom. Today, a biologist charts a family tree that spans 3 billion years. Beyond hide and feathers, he uncovers a startling unity that links us all to the beginnings of life on Earth. Twins are challenging our ideas about heredity. Perhaps our personality is shaped long before birth. A revolution has begun. Our world has been changed by a molecule called DNA. It unlocks an inner world and gives us the power to engineer life. This elegant spiral reveals how like begets like, how we develop and grow. It links us to the great chain of life. Yet it makes each of us unique. Join us now to explore a molecular universe. Journey through the intricate maze that is the geometry of life. Until he was 32, Mark Newman had no reason to suspect that his life story would be of any interest to science. He was adopted and raised in a middle-class family in New Jersey. As a child, he idolized John Wayne and loved the great outdoors. Perhaps the lure of adventure inspired him to become a fireman. Just 60 miles away, another fireman makes his rounds. Jerry Levy also loves firefighting. But he and Mark Newman have something else in common. Reunited after 32 years, they now know they are identical twins.
My parents had told me I was adopted. You know, they weren't my natural parents, so maybe there was some more family out there, but I really didn't think that much about it. Fine. That's what happened. I wasn't concerned about finding my real parents or if I had any, natural relatives, brothers or sisters. It didn't bother me, but in the back of my mind, there was, always felt like there was something missing out of my life. When I met my brother, that empty feeling I had, it was gone immediately. A little piece in the back of my mind saying that there's something missing in my life, it was filled. The fact that we were both firemen is a little spooky. We do have a saying, though, that, firemen aren't made. Fireman is born. So it's only natural. We're twins. We're identical twins. We're both firemen. We were born to be firemen.
Jerry and Mark are identical twins because they inherited identical genes. Their story challenges long-held notions about heredity. At the University of Minnesota, twins are helping scientists understand just how genes make us who we are.
Hi, Judy and Jill.
Hi.
How are you doing?
Good.
Good. Welcome to the lab.
Thank you.
Now, the first thing we want to do is to get some...
Judy Greer and Jill Freeman will undergo 6 days of testing that will include an extensive physical examination and a complete personality survey.
Get a measure of your weight.
Oh, keep going. A lightweight.
Yeah, that looks about right.
It's not surprising that on nearly every kind of physical test, their scores match.
Starting to work up a sweat?
Yeah.
What is intriguing is their psychological closeness.
Good effort. You better smile.
Normally, this would be attributed to a common family life. But even twins separated at birth often share the same mannerisms and style of dress, the same dreams and phobias. These uncanny similarities pique the interest of psychologist Thomas Bouchard.
Well, if somebody had come to me 10, 12 years ago, before I initiated this study and started examining these twins, and told me the results we have now, I wouldn't have believed it. My expectations were that there are more hills and valleys than there are flatlands. And by that I mean that I thought a good number of traits were largely environmental in origin, shaped by the family and the idiosyncratic environments that we experience, and that other traits are largely biological or genetic in influence. But our findings suggest that there's a kind of pervasive genetic influence that manifests itself in every kind of character we measure.
No, actually, one time I remember we were playing charades, and they put us on the same team, and I stood up and I read my little clue, and I yelled out, "Rocky and Bullwinkle," and she hadn't done one thing. Not one thing, and that was it. She just thought about it really hard. Yeah, it was really spooky.
The psychological profiles of twins are so close, they have forced scientists to accept the influence of genes on personality. Traits like leadership and shyness are largely inherited. Creativity, aggressiveness, and orderliness seem to be shaped by genes as much as by environment. And to some degree, genes produce extroverts and conformists.
We know virtually nothing about how you get from a gene to behavior. Our study says genes must be important, and therefore we should look at biological mechanisms. But boy, we really know very, very little about those mechanisms.
Twins offer us rare insight into the influence of genes on human behavior. They also confirm that genes determine the many traits that make each of us unique. Genes have endowed this child with red hair, blue eyes, and a mind of her own. But what is a gene? The answer lies hidden beyond the scope of the naked eye. The human universe, 100 trillion living cells. In each and every cell are all the instructions to make a human being. Safe in the nucleus is our genetic inheritance, the legacy of parents and forebears. These long molecules are called deoxyribonucleic acid, DNA. The structure of DNA resembles a ladder twisted into a spiral, known as the double helix. To form each rung of the ladder, 4 simple chemicals are precisely paired: adenine with thymine and cytosine with guanine. Dubbed A, T, C, and G, this is the 4-letter alphabet of the genetic code. Letter by letter, each of our genes is written along the double helix. A gene for eye color, another for blood type. Still others shape and define the brain. In all, 100,000 genes to create a human being. How is the genetic code inherited from one generation to the next? Because of its ingenious design, DNA can copy itself. First, the double helix unzips into 2 single strands. Along each strand, another molecule assembles each A with a T, each C with a G. Letter by letter, the missing half of each rung is recreated. The result, 2 identical copies of DNA. Now the molecules of DNA coil up into complex structures called chromosomes. The genes are ready to be passed on to the next generation. In time-lapse photography, a cell divides. Chromosomes are pulled apart. This is heredity at work.
Okay, here we go.
To welcome a new member of the human race is cause for celebration.
Oh, good.
Kisses, open your eyes.
Yeah. Oh.
Andrea?
For proud parents and relatives, a newborn baby is the precious tie that binds one generation to the next.
He's a sweet little brother.
Isn't it cute? Isn't that baby cute?
We delight in the discovery that the newborn has inherited mother's nose or father's eyes. We marvel at his perfect proportions, every pattern in place, each tiny hand and foot expertly sculpted. The miracle of life remains an ancient and profound mystery. In the 17th century, a Dutch biologist envisioned a miniature human curled within the head of the sperm. The idea that babies are conceived preformed and complete in every detail persisted for centuries. We now know that a human being begins life as a single cell. The sperms struggle toward the unfertilized egg. Each carries only half the genes needed to make a human being. At conception, their DNA entwines, and 2 family lines are united in one. Almost immediately, the egg begins to divide. Nothing more than the glimmer of chemicals animates these cells. For now, they are identical. Soon they will become brain and bone, heart and skin. The amazing transformation begins deep within each cell. Here is the machinery that will translate the language of DNA into a human being. On some elusive cue, a gene is switched on. A special molecule copies the gene letter by letter. Outside the nucleus, a tiny chemical factory decodes the genetic instructions and manufactures a protein, the raw material of life. With molecular precision, different genes choreograph the production of thousands of specialized proteins. Proteins now shape the destiny of each and every cell. This human embryo is about to be transformed. Within 2 weeks of fertilization, cells begin to congregate and build the shadowy foundations of a backbone. Some cells weave the network of the brain. Others fashion internal organs. At 4 weeks, a cluster of cells begins to pulse, and a tiny heart begins to beat. At 6 weeks, the embryo measures only half an inch, yet it bears the unmistakable imprint of human form. At 8 weeks, genes send out signals to lay the foundations of a human mind. The crescendo of some 25 trillion cells, exquisite and complex, still fascinates us all. At Columbia University in New York, a young scientist is exploring the mechanisms of development. Michael Levine is intrigued by a perplexing question. He knows that genes control development from single cell to complex organism. But how does the right gene turn on in the right cell at the right time? To find out, he turns to a simple creature, Drosophila, the common fruit fly. Fruit flies are ideal subjects. They breed prolifically. A new generation every 2 weeks.
So there's no problem with the road.
Early on, researchers noticed mutations in the fruit fly, naturally occurring variations in body size, wing shape, and color. Mutations are caused by changes in the fruit fly's genes. Scientists can also create mutations of their own. First, DNA is extracted from a fruit fly and chemically altered. With a needle thinner than a human hair, Levine injects the man-made gene into an embryo only 2 hours old. If a mutation occurs, it may help him understand the role of this gene in development. Simple mutations help identify genes that control wing shape and eye color. But a series of bizarre mutations would hint at more powerful genes.
Over the course of the many years of creating mutations in different Drosophila genes, a rather spectacular class of mutations, called homeotic mutations, have been identified that have important implications as to how flies and perhaps other organisms develop. In general, mutations in homeotic genes cause the formation, in a sense, of the right structure in the wrong place. For example, here, this is a normal fly head that has a pair of eyes and a pair of antennae, plus various mouth structures here. In the homeotic mutation antennapedia, the antennae are transformed into perfectly shaped legs, as you can see here, much longer structures than the normal antennae.
The transformations that led to extra sets of legs or a second pair of wings would have profound implications.
The occurrence of these very spectacular mutations immediately suggested that not all genes are equal in controlling developmental processes, and suggested that these genes are master switches that really control development in a hierarchical way. Namely, they influence the activities of other genes, of subservient genes, which in turn execute the will of the master switch.
Shortly after fertilization, master genes are activated. They trigger more specialized genes that transform cells into wing or leg or antenna. Within hours, the fate of every cell will be sealed. Levine has isolated a gene that designs the fruit fly's basic body plan. Now he watches the drama unfold. 2.5 hours after fertilization, an embryo appears undefined and amorphous. Only 20 minutes later, a master gene creates order from chaos. Head and tail are defined. Within minutes, the major body segments emerge. The architecture of a fruit fly is in place.
It's as if the regulatory genes were like conductors that determine when certain musicians play and when others don't. By analogy, the regulatory genes determine exactly when many thousands of genes are turned on and off, just as a conductor determines when musicians will play certain passages and others won't. And without the regulatory genes, without the conductors, there's chaos.
And so it is the exquisite orchestration of genes that transforms a single cell into a complex organism. Levine believes that similar master genes may be at work in higher organisms, including humans. The simple fruit fly yields a clue to one of life's enduring mysteries. Each year, some 3 million people visit the San Diego Zoo. Our fascination with other creatures begins in childhood. Perhaps it is ingrained in us. We have long wondered who is related to whom. Similarities of anatomy and behavior are telling, but the forces of evolution can trick us with subtleties that confound the human eye. Until recently, some of nature's most intriguing family ties were beyond our grasp. Russell Doolittle of the University of California, San Diego, is tracing the thread of evolution. He calls himself an archaeologist, but he studies no bones, digs no fossils. His tools are the computer and molecules. Like a cryptologist who deciphers an ancient and lost language, Doolittle uses the genetic code as a biological Rosetta Stone. For him, the history of life is written in the molecules of DNA.
These are extraordinary times in the field of biology. The main reason being that there is this deluge of information coming in from what's called DNA sequencing. DNA sequencing is where you study life in organisms by finding out the genetic blueprint. It comes out in the form of a Morse coding, just millions and millions of bits of 4 little dots and dashes. They're called A's and G's and C's and T's. And out of that, we can read the whole story, not only of a particular organism, but where we all came from. We're reconstructing 3.5 billion years of evolution on the Earth.
To read DNA, the molecules are first broken into fragments. Stained and tagged with a radioactive dye, they are then injected into what technicians call an electrophoresis gel. A powerful electric current pulls the fragments through the gel. Separated according to size, they leave a ghostly pattern behind. When the gel is exposed to X-ray film, a telltale pattern appears. Each band on the film corresponds to a letter along the double helix. Letter by letter, a sequence of DNA is painstakingly logged into the computer. The challenge for Doolittle is to translate DNA's code into the language of proteins. He will then compare the sequences of proteins to see how 2 species are related.
Well, one of the things that's being revealed by all of this DNA sequence information that's flooding in is the amazing unity of all the living organisms on the Earth. I think that there are a lot of scientists who are surprised at how similar many of our enzymes are turning out to be, many of our proteins and our enzymes in general, to bacterial forms, for example. I know that I was surprised to find that if we took a simple enzyme that's involved every time we burn our sugar, and if we looked at the sequence from the DNA and the corresponding protein in the enzyme, and we compared us with a bacterium like E. coli, which is found in our gut, found out that, in fact, well, we'll bring it up on the screen here. Now, if we look on the screen, we see that the human enzyme has 249 amino acids in it. The E. coli bacterium has 255. Similar, but not quite the same. But we look in closely, and we see that 115 of them are exactly the same, 46% identical. A few little places where there's some gaps, but we expect that sort of wear and tear during the course of 2 or 3 billion years of evolution. We'll move it just a quick step further. We'll go now to a chicken enzyme. 223 identities out of 249. Its sequence is exactly the same length as the enzyme that you and I have. No gobbling chicken has this same enzyme, and it's so similar, 89.5% identical. If we move up to a mammal, move into a rabbit, for example, we can bring that one up on the screen now. Rabbit in here, it says. Holy cow, it's hard to tell rabbits from people here. It's 244 identities out of 248. That's how many there are in the whole enzyme. A whopping 98% identity.
Face to face with a chimpanzee, we must wonder if we are gazing at a distant relative. The great apes stare back with equal curiosity. A new family tree is taking us all by surprise.
The first protein to be sequenced of any length was determined both from a human and shortly thereafter from a chimp. And it was a piece of a hemoglobin. And the amazing thing is, through all of 146 amino acids, the chimp had exactly the same as the human. No difference. Really surprising. So hemoglobin, turns out, has 2 different chains that go into a molecule. The other chain, which is called the A chain, was sequenced. That only has 141 amino acids, but there, once again, all 141, exactly right on, the same protein. A third protein was looked at. It's called a cytochrome. Human, 104 amino acids. Chimpanzee, 104. Every single one of them the same. Now, in fact, I was called by some people to look at some proteins that we worked on that were known to be very variable from species to species. And so we pulled them out, looked at them. They were identical also. The rumor began to go around among the circuit that, in fact, all the differences between chimps and people were going to turn out to be cultural. Turned out wasn't true. The 5th protein that was looked at happened to have some 200 and some odd amino acids. There was indeed a difference. Phew, thank heavens they really, they are different from us.
Other studies confirm our DNA is nearly 99% identical to the chimpanzees. A mere 1% difference accounts for human language and culture, art and science, and for the curiosity to ponder our ancient origins.
What's emerging from all this DNA sequence data is an enlarged picture of how molecular evolution has come to be. We've always, as biologists, thought that life must have had a single start on this earth, but now all the DNA sequencing data are certainly proving it. In a sense, we have all become molecular historians. We can figure out the way things were 1 billion years ago, 2 billion years ago. There are little molecular fossils here and there, time capsules, if you would like, that are telling us the way events used to be. What's coming out of this is how we all got to be the way we are today, and that's certainly what I would like to find out.
A genetic thread links us to the beginnings of life on Earth. In the first cells were tiny fragments of DNA that allowed them to multiply and fill the primordial seas. Slowly, they gave rise to more complex forms, varied, intricate, and unique. Perhaps once in a million times, as DNA copies itself, it makes a mistake. Over time, simple errors can lead to profound changes. From these humble beginnings, all living things evolved. But in a human being, a simple error can be devastating. There are 3 billion rungs in the ladder of human DNA. A mistake in a single rung can make the difference between life and death. Lake Maracaibo, Venezuela. Here, 100 years ago, a woman named Maria Concepcion Soto died of a neurological disorder called Huntington's disease. Today, her descendants carry her deadly gene. Huntington's disease usually strikes in midlife. It causes slow deterioration of the brain and nervous system. There is no treatment, no cure. In New York City, Nancy Wexler is a clinical psychologist. Her mother died of Huntington's.
When my mother was diagnosed, my father took my sister and me into his room and said, your mother has this illness, it's fatal, and each of you has a 50-50 risk of inheriting it. And I think from that day, really my life changed enormously.
In 1981, Wexler went to Venezuela to track the defective gene.
¿Tú conoces Ángel Jiménez Flores Enciarte que vive aquí? Ángel Guerra.
The villages along Lake Maracaibo contain the highest concentration of Huntington's disease in the world.
Cileida Margarita. Cileida? Margarita.
Wexler is charting a family tree to establish the pattern of inheritance. Eventually, it will include over 2,000 people. Half stand a 50-50 chance of developing the disease. Many are children.
You want to sit down?
¿Cómo es tu nombre? ¿Cómo te llamas?
To understand Huntington's, scientists must first find the defective gene. It is an anxious moment for young Beatriz Soto as a sample of her blood is taken. Wexler will collect over 250 samples. Somewhere in the DNA is the answer. At Massachusetts General Hospital in Boston, Wexler's samples are turned over to biologist James Gozella. Looking for a gene is like looking for a needle in a haystack. But Gozella had a plan. He would compare the DNA to find a marker, a distinctive pattern that is always inherited with the disease. The search might have taken decades. Incredibly, on the 12th try, he found a marker.
This is an example of typing people with the marker linked to Huntington's disease. Each lane here represents the DNA from a different member of the very large Venezuelan family that Nancy Wexler has collected. And we can correlate the patterns shown here with the relationships of these people in the very large Venezuelan family as to who's parents of the various children, who's the mother, who's the father. We then look at those patterns and see which one correlates in inheritance with the disease gene. In this particular case, it's denoted by the presence of this lower band here underneath a more common band found in everyone. In this case, we have an affected woman who has this band, whose husband is unaffected and doesn't have it. They had 4 children. The first 2 children do not have the band, and they, in fact, do not have Huntington's disease. The next 2 children do have the band, and they both have Huntington's disease.
For Nancy Wexler and the people of Lake Maracaibo, the discovery of the marker is a mixed blessing. Eventually, it will lead scientists to the gene for Huntington's, and hopefully to a cure.
When you look at those children and you know that some of those children have to be carrying this fatal gene, and you know what's going to happen to them. You see it. You go in and you visit their mothers and fathers dying in bed, and you know that that's going to happen to them. It's absolutely horrifying. And I know before we found the marker that sometimes I would feel holding a little girl who was dying of Huntington's, and I wanted to shake her as if I could just shake that gene out of her. I could see what was in it. And I would know, where are you and what are you doing to her? And it keeps you possessed. I think we work like madmen down there a lot of times, because you know that if you get there that much sooner, 1 year sooner, that maybe you're saving lives.
It is a race against time to hunt down the deadly gene before a new generation falls victim. But for the children of Lake Maracaibo, there is now hope. Six 6,000 years ago, humans first tamed a stocky wild horse. Since then, we have shaped the animal to suit our fancy. The thoroughbred, the crowning achievement of centuries of selective breeding. But creating a champion is an imprecise science. Breeder Buddy Johnston has been at it for 30 years.
We're very excited about this new baby. His father was second in the Kentucky Derby, won over $1 million. His mother was one of the best race mares in America. And now with a little luck, maybe we've got one that can get us to the winner's circle. There you go. He could be a good one.
Even with good odds, a winner is still a long shot. Today we can manipulate life as never before. A revolution has begun. With chemical tools to cut and splice DNA, we can move genes from one organism to another, or recombine DNA from two different species. With recombinant DNA technology, researchers can now convert the simplest forms of life into living chemical factories. Inside a bacterium are small circular loops of DNA. Through intricate chemical surgery, a loop is cut open and a human gene inserted. The bacterium cannot distinguish the human DNA from its own. It will reproduce as usual, doubling its number every 20 minutes. Within hours, millions of microscopic factories churn out human protein. On an industrial scale, genetically engineered bacteria are grown in 10,000-gallon vats. From the simplest of organisms, we reap a harvest that can save human lives. In 1982, human insulin became the first genetically engineered drug to reach the market. Since then, we have used recombinant DNA technology to develop improved vaccines and precise diagnostic tools. We are custom designing molecules to fight cancer, and we can now treat a genetic disease that has plagued us for centuries. Consider the royal disease. Queen Victoria lived to the ripe old age of 82, but she carried a gene for hemophilia, a bleeding disorder that afflicts only men. Victoria's gene made its way into several of the royal families of Europe. Her granddaughter Alexandra married Tsar Nicholas of Russia. Their son Alexis inherited the disease. Hemophilia is caused by a mutation in the gene for blood clotting. Victims suffer internal bleeding and are crippled by their own blood. There was no treatment for Alexis. It is said that the royal palace echoed with his cries. George McCoy's family has also been shattered by hemophilia. The defective gene was unknowingly passed on to him by his mother. George was born in 1947, the youngest of 4 children. One of his sisters is a carrier of the disease. His older brother Richard succumbed to hemophilia at age 29.
In 1947, there was very little known about hemophilia, and there wasn't a lot of treatment. But when I went to first grade, our family doctor advised that I go on crutches to protect me from the other children in case of an accident. And that was when I began to realize that I was very different, and of course that other kids didn't go to the hospital and have to stay for days when something bad or dangerous would happen. By the time that I hit high school, I was on a roll. I was doing fine academically and active in a lot of clubs, and became student council president my senior year. And I went away to college, and those years were also successful. It was when I left college and got married and expected to go out and find a job that I really encountered a lot of difficulty, and probably the biggest time in my life of being limited by hemophilia came at that time. And I would go for an interview, and one place would tell me, no, we hired a person with hemophilia here, and we'll never make that mistake again. I was very depressed and wouldn't leave the house, kept the blinds pulled, wouldn't answer the phone. And although I had a lot of love and support from family and friends, I still was very depressed. What turned it around for me was discovering the factor VIII, and that was the clotting protein that I was missing.
Hemophiliacs lack a crucial blood protein called factor VIII. In the 1960s, researchers found a way to extract it from human blood. For the first time, hemophiliacs could treat themselves. Before, the only therapy had been massive transfusions and extended hospital stays.
Because of the factor VIII and my ability to use it to control the bleeding, I could drive a car, I could walk around on a campus, I could have a social life, and eventually hold a job and be there every day. I could be happy. I could be a human being, basically.
But the miracle drug had its dark side. Because factor VIII is derived from human blood, it can carry viruses for hepatitis and AIDS. For hemophiliacs, it is like playing Russian roulette. In 1987, George received a phone call from his doctor at the University of North Carolina School of Medicine.
Morning, George. How are you?
All right, how are you?
Okay.
Dr. Gilbert White asked George if he would volunteer to be the first trial patient for a new form of factor VIII. Because this protein is produced through recombinant DNA technology, it is free of viral contamination. George agreed and became a pioneer in medical history.
Have you had any bleeds in the last month?
Had one problem with my knee, and that took a couple of infusions because I had let it go just a little bit longer than I should have. I thought it was arthritis. Other than that, things have been pretty good.
And the factor VIII still goes into solution as quickly as it has been.
Thanks to George, this new drug may soon help many hemophiliacs whose lives have been torn by suffering and fear.
I think to me the most exciting part of it is that the engineers who developed this have told me that they feel that what they've learned from this research may eventually lead to a cure for hemophilia through genetic engineering.
The search for that cure is underway in a laboratory in Seattle. Scientists are developing a revolutionary form of genetic therapy. Still in the experimental stages, it has only been tested on mice. Dusty Miller wants to beat genetic disease at its own game. At the Hutchinson Cancer Research Center, his goal is to cure hemophilia. He wants to transplant a corrective gene into a patient's cells.
I think we're into a new era as far as treatment of disease. Before, you could treat diseases with drugs. You could treat them with things like eyeglasses or hearing aids. diseases of hearing. But now we're into an era where we can say we know what the specific defect is in an individual, and we'd like to correct that for the rest of that individual's life on a fundamental basis. That is, replace or repair the gene that's defective in that individual or in some proportion of that individual's cells. Now, this isn't a hereditary type process. The individual won't pass that on to his or her descendants, but it will at least make that person live a normal life.
The challenge? To get the right gene into millions of cells. With colleague Theo Palmer, Miller is harnessing one of man's oldest enemies, the retrovirus.
I guess I'd call retrovirus more nature's genetic bullet. So it's an organism that's designed to go and infect a cell and deposit its genetic information into the chromosomes of the recipient cell. We've taken that natural ability to infect a cell and then deposit the genetic material and modified it so that we can transfer our own genes.
First, Palmer will strip the retrovirus of its harmful genes. Next, he will replace the harmful genes with a human gene that will cure hemophilia. Now Palmer infects a cell culture with the engineered virus. As viruses dock on the cell surface, new genes are delivered. These genetic bullets head straight for the nucleus of the cell. Here, the corrective gene knits itself into the DNA and becomes a permanent transplant. An old enemy becomes an ally. The altered cells are grown into a skin graft that will produce the blood clotting factor. In tests on mice, these grafts secrete the protein that hemophiliacs lack. The transplanted genes would alleviate the patient's need for lifelong treatment, but would not be inherited by their children. Still, these same techniques offer great hope to victims of many other genetic diseases. Few doubt it will transform the future of medicine. As we harness the molecules of life, we reshape the world we live in. Biotechnology promises great bounty. Crops resistant to frost and disease, improved strains of livestock, self-fertilizing plants that double their yield, and special microbes to process toxins and clean the environment. But as we peer into the microcosm of the cell, we come face to face with a great dilemma. These incredible tools may lead us to the brink of manipulating our own genetic inheritance. How far will we take this technology? How well will we use it? Mankind may soon confront its greatest challenge. If one event is to mark the beginning of the biological revolution, it is the discovery 35 years ago of the structure of DNA. James Watson was a postdoctoral fellow at England's Cambridge University. In 1953, he and colleague Francis Crick proposed the double helix structure of DNA. They would stun the scientific world. With their simple tinker-toy model, they revealed the form and function of the master molecule of life. For their achievement, they would win a Nobel Prize.
The formulation of the double helical structure of the deoxyribonucleic acid.
The profound implications of their discovery had already begun to change the study of life forever.
When we found it, we had no idea that, say, even within our lifetimes, we were going to get anywhere up to where we are now. The concept that we'd actually know the sequence of a given gene, that we would be able to clone a gene, that would have been science fiction for us then. But due to these sort of technological advances, we can do things that no one dreamed we could have back then.
Perhaps the most profound exploration into the molecules of life is the search for who we are. Unprecedented self-knowledge is now within our grasp. Today it is the subject of public debate. In 1987, the United States Senate began hearings to consider a monumental plan called the Human Genome Project. Its goal, to decipher the complete genetic code, or genome, of a human being. The project will require the resources of national laboratories, universities, and private industry.
What we really would like to do is to establish that the mapping of the human genome on an expedited track is a goal, and why, and if it is...
The Human Genome Project is daunting because of its scale, similar perhaps to putting a man on the moon. There are an estimated 100,000 genes in the human genome. To date, we have identified only 1%. As we begin to locate all of our genes, we will pinpoint the cause of thousands of known genetic diseases, the first step toward a cure. By decoding the sequence of some 3 billion rungs of human DNA, we will finally have the complete blueprint of a human being. It will inaugurate a new age of molecular medicine. A leading proponent is Nobel laureate Walter Gilbert of Harvard University.
The Human Genome Project flows out of a number of advances in biology. There are moments in the history of science, like the discovery of DNA, when a particular discovery informs or changes the way in which we look at the world. The discovery of DNA told us how the genetic information was passed from parent to child. The discovery of the genetic code told us how that information could be read out. The discoveries of evolution have told us how we fit in the great chain of life from the beginning of the Earth. The Human Genome Project is a working out of those particular ideas. It does represent a particular moment in history in which we will first know all there is to know about the human being from the genetic side.
With current techniques, it would take a single scientist some 30,000 years to decipher all the letters in the human genome. The success of the project depends on the development of high-speed technology. Lasers will penetrate the world of the cell to sort its molecules. Automated scanners will map the landscape of each human gene. And powerful computers will catalog and store the vast amount of information. Ultimately, the book of mankind will fill an encyclopedia of 1,000 volumes.
At some point, 10 years from now, it's going to be possible to go to that 1,000-page book, the 1,000 copies of that book, and say, here is an entire human being. In fact, one's going to have a human being almost on a compact disk, a computer disk of information. You'll be able to hold it in your hand and say, in a sense, this is a human being.
Our job won't be over at all when we have got the sequence, because then we'll have the book and we'll want to read it. And we know some of the language, but we don't know all of it. And it's as if you have a very difficult poem, and you're going to try and get all the sense out of it. And I think it'll take us thousands of years to really understand that, the message. But that's a relatively short period of time if you consider the length of human evolution. And I think it's extraordinary that human beings will have evolved and have brains remarkable enough so they can actually work out what we are.
We are all the product of the molecular geometry at the heart of life. The exploration of DNA is beginning to answer our most perplexing questions. How a single cell becomes a human being. we develop and grow. How a simple code spells out the great diversity of life on Earth. How we are all connected to the past, to each other, to the future. In the midst of this great adventure, we may discover what makes us unique, human. It is a voyage that has just begun.